Classification of breast cancer. Breast cancer is classified into several types based on where it begins in the breast, how much it has grown or spread, and how it behaves. The aim of classification is 1. Diagnosis, 2. Prognosis, 3. Prediction. Classification type of breast cancer is majorly classified as to invasive breast cancer and non-invasive breast cancer. Invasive breast cancer means that it has spread from the breast ducts or lobules into the surrounding tissues. Types of invasive breast cancer, we have 2: invasive ductal carcinoma and invasive lobular carcinoma. Invasive ductal carcinoma is also known as infiltrative ductal carcinoma. Cancer cells that begin in the milk duct spread to other parts of the breast tissue. These account for majority of all breast cancers. Invasive breast cancer that begins in the lobule, that the milk producing glands in the breast, it is the second most common type of breast cancer. You can see this is a picture of ductal carcinoma. This you can see here is localized. These are localized ductal carcinoma from the ducts, milk ducts. And here we have an invasive cancer, invasive ductal carcinoma. It has spread, it has invaded the nearby tissues. And here we also have the lobules, the milk producing glands. These are lobules. These are lobule. So this is localized. It's within the lobule. And this has infiltrated to other tissue, nearby tissues. That's an invasive cancer. Non-invasive breast cancers, also known as precancers. Non-invasive means that the cancer has not spread beyond the breast tissue where it began. Non-invasive breast cancers are also known as precancers. There are 2 types of non-invasive breast cancer, that is ductal carcinoma in situ and lobular carcinoma in situ. Ductal carcinoma in situ, it means this type of breast cancer cell that has not spread beyond the milk ducts where it began. Ductal carcinoma in situ is not life-threatening, but is a precursor to invasive breast cancer and increases the risk of developing it later in life. Lobular carcinoma in situ, remember the lobules are the milk-producing glands at the end of the breast ducts. This is a benign breast condition, but it does not increase the risk of developing breast cancer. We also have another form of classification called molecular classification. Our understanding of breast cancer has changed as a result of discovery of 4 molecular subtypes by gene expression profiling, namely Lumina A, Lumina B, human epidermal growth factor receptor 2, and BASAL-like. This classification is based on the presence of estrogen, progesterone, and human epidermal growth factor receptors. We start with Lumina A. This is the most common subtype. It expresses hormonal receptor for estrogen and either progesterone or not, but it does not express human epidermal growth factor. Lumina A breast cancer has a slower growth rate, is less aggressive, and typically produces better results. When you go to Lumina B, it expresses hormonal receptor, estrogen. human epidermal growth factor, that is HER2, and either or not receptors of progesterone. Compared to Lumina A, Lumina B is graded higher than A. Lumina B is more aggressive than Lumina A, has poorer outcome compared to Lumina A, but it responds well to HER2 targeted therapy. The third one is HER2 positive breast cancer. This type of breast cancer only tests positive for human epidermal growth factor receptor 2. It does not express hormones receptor for either progesterone or estrogen. What is HER2? Human epidermal growth factor is a protein that promotes growth of breast cancers. HER2 positive breast cancer have higher than normal levels of HER2. These cancers grow and spread faster than HER2 negative breast cancer, but they are more likely to respond better to treatment with drugs that target HER2 proteins. How do we test for HER2 status? The following tests are used to test HER2 status. We have immunohistochemistry, that HHC test, and fluorescence in situ hybridization, that is FISH test. BASAL-like. The fourth one is called BASAL-like, the triple negative breast cancer. Triple negative. This means that all the receptors are negative. They lack estrogen receptors, they lack progesterone receptors, and the HER2 receptors. Characteristics of this: it grows and spreads faster than all breast cancer types. It's aggressive. It has fewer treatment options, and it has the worst prognosis compared to all breast cancer types. In summary, this is what we have been talking about: the molecular subtypes of breast cancer, Lumina A, Lumina B, HER2, and BASAL-like. And we are looking at the common ones, the three common types of receptors: estrogen receptors, progesterone receptors, and HER2 receptors. Lumina A and B, estrogen receptors are positive, but when it comes to progesterone, can be either positive or negative. For Lumina B, the major difference between A and B is the HER2 receptors which are present in Lumina B. When you go to HER2 type of breast cancer, estrogen and progesterone are negative, but the HER2 receptors are present. The last one is the BASAL-like, the triple negative breast cancer. Triple means triple negative, that they are not present. All the receptors are not present, and that's why we normally say it has poor prognosis. And those are my reference. Thank you, and kindly subscribe and share.